The evolutionary model holds that living things, like myself, share ancestry with other living things. The creation model holds that that is not the case, where living things like myself would not be related to anything that isn't human, that they would be completely unrelated, 100%. In intelligent design, complexity cannot evolve in stages. And so therefore, one of the ways to test these three models, and in science, that's what we do. We come up with alternate ideas, new ideas based on new observations, et cetera, and then decide how could they be tested. Well, one could then study embryos and see if embryonic evidence supports any of these models. One of the things which results from a study of embryos is the conclusion that embryos certainly are not miniature adults. So when the human brain forms in embryos, or human bones, like the occipital bones, the temporal bones, or the kidneys, etc., they could form as miniature versions of the structures found in adults, and then just slowly take on the adult form. And if the creation model is correct, then that would be expected, that these human embryonic structures who only appeared as precursors to the human form would then have a simple human form and then would simply become larger. But that is 100% not what is observed. In embryos, human embryos have the features of which they share with other embryos which aren't human, and many human anatomical traits then gradually take on this human quality after appearing with the characteristics of species which aren't human. So let me go through some examples. Embryos not only have some of their cells form organ systems, like a brain, a heart, a skeletal system, etc., but also some then form membranes around the embryo. So there's the yolk sac, as you can see in some of these organisms. And then there are several others. These extra embryonic membranes can be adapted for different purposes. So in fish, for example, we see that some can actually have a primitive placenta form. But if you were to say look at this egg-laying lizard, you would see an extra embryonic membrane, the yolk sac. By extra embryonic, I mean that these are the cells of the embryo, so that not all of the embryo then makes up, brain and bone and muscle, but also some form this bag. Some of the embryo's cells form this bag, that's the amnion. Some of the embryo cells form this bag, the allantois, and some of the embryo cells form this bag, the chorion. So the early reptiles, the early amniotes, then had 4 extra embryonic membranes around themselves: an amnion, hence the name, a chorion, a yolk sac, and an allantois. And these extra embryonic membranes come in handy when an embryo is developing in an egg. But here you can see a live-bearing lizard, and while some lizards lay eggs, others give live birth. And you'll notice that it has the same membranes. Now, because live birth has evolved multiple times in related lizards, it's obvious then that the extra embryonic membranes, which were adapted to developing inside an egg, could be then adapted to developing inside the mother as well. If we were then to look at other mammal embryos, one would see the exact same 4 extra embryonic membranes: the chorion, the amnion, the yolk sac, and the allantois. even in humans. Now, obviously, human embryos have an amnion because we develop in an amniotic fluid, and this helps to absorb shock. Obviously, we develop inside a chorion because this is what forms the fetal part of the placenta. But the yolk sac and the allantois, they don't have their ancestral function. The yolk sac is not storing food. The nourishment for an embryo and fetus comes from the mother's bloodstream and the placenta. The allantois does not store nitrogenous waste. It's the mother's kidneys, which are involved in the removal of waste. And so human embryos form a yolk sac and an allantois, the same extra embryonic membranes as formed in egg-laying animals, even though they aren't as needed. Now you might point out, well, don't some blood cells develop in the yolk sac at first before the liver takes over making blood cells? Yes, but they don't have to. I mean, the liver can make blood cells, as can the skeleton, as it does after birth. There's no reason that a yolk sac is needed for blood cell production. There's no reason that the stem cells, which will end up in the ovaries and testes, have to begin in the boundary between the yolk sac and the allantois outside the body of the embryo. And so the fact that human embryos have the same extra embryonic membranes as other animals, including egg-laying animals, is support of a shared common ancestry. If humans were 100% unrelated to egg-laying animals, there would be no reason that a yolk sac and an allantois would be needed. The human heart has a specific shape and form and number of chambers, which is different from, say, a lamprey, a simple vertebrate, where there is an atrium, I'm sorry, there is a sinus venosus, there is an atrium, a ventricle, and a truncus or a conus arteriosus, one atrium, one ventricle, and then these two additional chambers. Now, when one looks at the developing heart in a human embryo, one finds a sinus venosus, a single atrium, a single ventricle, a single truncus, or conus arteriosus. And so the human heart begins its embryonic development not as a miniature human heart, but as something that looks like a fish heart with the same chambers. There is no reason that that would have to be the case, and that is unexpected if humans are 100% unrelated to fish, that our heart would begin in a form not like the heart, but rather like a fish heart. And then the same thing when we consider, I'm just making sure I didn't skip one. When we consider other parts of the skeleton as well. So for example, as an adult, I have one occipital bone. I have a pair of temporal bones, I have a sphenoid bone, I have a frontal bone, etc. These are the human bones. But embryos don't have miniature versions of human bones, nor do things like reptiles. Reptiles don't have an occipital bone. They have lots of little bones in the occipital region, which we could name a basio-occipital, exo-occipitals, post parietal, tabulars, etc. And fossils indicate that these small bones gradually fuse together to form the single occipital bone in primitive mammals. And the same would be true of temporal bones, that reptiles don't have a temporal bone. They have a squamosal bone, a petrosal bone, these other little bones which fuse together. Same with the frontal bones. There was a pair of those, etc. Fine. But if humans are 100% unrelated to these other organisms, one could expect then that the human embryos begin with miniature versions of human bones. But what we see is that human embryonic development of skull bones mirrors the fossil progression from reptilian bones to mammalian bones, where little ossification centers fuse together. So human embryos have two frontal bones, two vomers, two halves of the mandible, just as did more primitive organisms. The incisors were held in a premaxillary bone, which would then fuse to the maxillary. The single temporal bone or the single occipital bone is in human embryos represented by separate ossification centers which fuse. Not only the separate ossification centers, but the same ossification centers as we see in, say, reptiles. So just as reptiles have an basooccipital and exooccipitals, so did I as a fetus, but these then just fused together to make the my occipital bone. My lower jaw had a bar which was in the same region as other jaw bones in more primitive animals. Two of my inner ear bones began in association with jaw and then migrated to, the middle ear region, just as the fossil record suggests. And so the skull bones of fetuses don't form as miniature versions of the adult human bones, but they form in a way that we would expect if humans share a common ancestry with other animals. The same would apply to the vertebrae. In more primitive animals, it is a notochord, which is the primary support rod along the back. In early vertebrates, the vertebrae start as little pieces of cartilage above and later below the notochord, which gradually get bigger, get converted to bone, and then fuse. Well, humans still form a notochord, even though we do not use a notochord as our support rod. We have a vertebral column. Nevertheless, we form a continuous notochord as our first support. And then little pieces of vertebrae form around it and then gradually grow and fuse, just as the fossil record suggested that vertebrae evolved. And so our vertebral column does not begin looking like miniature human vertebrae, but rather forms in the same way that we would expect if we are related to more primitive animals in which the vertebral column formed very gradually through intermediate steps. Many other parts of the skeleton are one human hip bone, forms from the same three separate bones, the ilium, the ischium, and the pubis, that we see in other animals. Our clavicle forms from fusion of separate elements that even form through different types of ossification. Part of the clavicle forms as dermal bone through intermembranous ossification, part as endochondral bone. Our transverse processes in our cervical foramen, these result from the remains of cervical ribs. More primitive animals had cervical ribs in their neck vertebrae, so did we, but then they fused to the neck vertebrae. So our skeletal system does not form as a miniature version of the human skeleton, but bears resemblance to other animals and how the skeletal systems forms in them. Now, fish obviously have gills, which is very important. This is where the water that comes in the mouth exits their body, gas exchange occurs. So the slits in their throat are important for them to breathe. They have these big pharyngeal arches, pharyngeal slits between them. Branches of a ventral aorta go to each of these to perform gas exchange. They have nerves going to each of these. This is very important. But one finds these in animals which don't have gills as well, such as these images from a chicken, this drawing from a human, etc. We had pharyngeal arches. Here's a pig embryo. Notice the pharyngeal arches there. There is no reason for a chicken, a pig, a human to make pharyngeal arches with slits between them, with branches of the ventral aorta going to each, because this then has to be reworked. But nevertheless, we develop our throat region as if we are planning on putting gills there, only to subsequently rework the area, which, as I'll get to in just a little bit, leaves us vulnerable to birth defects, and I'll get back to birth defects presently. And so our throat region does not support the conclusion that we are unrelated to other organisms, but rather that we share with other organisms. Now that throat region is also then very important in blood vessels. When one considers the arteries and veins that humans have as adults, this is not what forms as embryos. We modify our first blood vessels, which have more of a fish pattern. So when we were embryos, our early blood vessels are ones which remind us of those vessels found in fish. Let me go back to that one. So fish have a very clear organization of the blood vessels that go to the gills. We begin with this same ventral aorta sending branches to our pharyngeal arches before they reunite to form a dorsal aorta. But these then are reworked to find to form the blood vessels which we have. We still number them developmentally. You know, here is the derivative of arch 3, arch 4, arch 6. The other ones form and then deteriorate. So even though our blood vessels, like, you know, the carotids, the pulmonary trunk and artery, even though that's different from what forms in fish, nevertheless, we begin with a fish circulatory pattern, which then gets modified. Our kidneys in adult humans are different from, say, those of fish. Fish have a pronephros region, then a mesonephros, and then finally a metanephros, which is homologous to the part that we have. But as embryos, we form fish kidneys. We form a pronephros, a mesonephros, a metanephros all down our backs. The pronephros and mesonephros, they then are broken down, and the metanephros, which is in the pelvic region, this then has to ascend because the pelvic region is not where the kidneys are kept as adults. So this has to then ascend to the position that it will acquire later. And so our urinary system does not form as a miniature version of the human urinary system, but rather as something similar to those we find in other animals. It is only then modified to form the structures which we have. In the same way, just as the kidneys are beginning lower and will have to then rise, the gonad here in red starts high. In both males and females, it starts in the abdominal region. In females, it must descend to the iliac, this is not the situation in humans, but animals, say fish, amphibians, reptiles, birds. One finds a structure known as the cloaca. The cloaca here in purple, this is the shared region for the large intestine to empty. In other words, it includes the anus. It includes the urethra, which is how the urinary system empties. And then in females, it would include the vagina as well. So this is the shared opening for the digestive, urinary, and reproductive tracts. Now, obviously, we have separate openings, but rather that which we associate with other animals. And we could go on and on. I'm going to go through some slides, but this is important. You know, you might say, well, you know, why? You know, who cares in essence? And the reason that we do is that mistakes in embryonic development can lead to birth defects and to lead to miscarriages. It's, you know, we could argue perhaps about percentages, but perhaps about 30% of human conceptions do not make it to live birth. Many of the infants which are born are born with developmental defects, which have to then be corrected. So birth defects and miscarriage, these are serious problems for a developing embryo to avoid. Extra steps increase the risk of these. If an embryo is going to form structures which it does not need, and then has to undo and form new ones. These are simply extra steps, which then open the possibility for birth defects. So, for example, most of the land vertebrates possess prominent tails. Human embryos develop prominent tails, which occasionally persists as external tails, which then even can include muscle, which then have to be surgically removed at birth. The pharyngeal arches, which form in chicken embryos, pig embryos, and human embryos, then sometimes persist, and we can have conditions in our throats where we have clefts which have to be surgically closed, or fistulas where parts of the pharyngeal arch is now causing a cyst. And so there can be issues there. The notochord can persist in some individuals. Some individuals may actually even then have cancer of the notochord. The heart forms as a modified version of a fish heart, and as it then divides from that ancestral condition to the derived condition, there are a number of birth defects which are possible, some quite serious. As I mentioned, we start off with all of these blood vessels which we don't have as adults, cardinal veins, those aortic arches, etc. And there can be abnormal blood vessels which form in humans as a result. This cloaca, which forms, then has to subdivide. But it can subdivide abnormally. And so in some infants, the large intestine may empty into the vagina or into the urethra, and this would then have to be corrected. Humans start off with these paddle-like limbs, and in some individuals, that is as far as they progress. Notice here the kidney, the mesonephros, goes up to the armpits. All right, and so this then has to degenerate. Now, some individuals, as the kidneys are forming high, and are low, and the gonads are forming high, and they have to switch places. In some individuals, the kidney stays in the pelvis, where the two fuse on their way as they rise and form a horseshoe kidney. So there can be birth defects there. Or there could be a birth defect where someone's gonad remains in the abdomen, an abdominal testis, for example, which would never be fertile. The germ cells start here. Even though they're going to end up in either the ovaries or the testes, they start outside the body, between, say, the yolk sac and the allantois, and they then have to migrate and find the gonads. Now, in some individuals, they never do, and so some individuals are infertile because these primordial germ cells never appropriately migrated from the place where they originated into the gonads. The uterus and vagina form from a pair of tubes which fuse, but the fusion of these tubes can leave a septum in the vagina or in the uterus, or a Y-shaped uterus, which has to be corrected. Mammals develop mammary glands, but can vary where they do so. And so mammal embryos develop a mammary ridge. And so different species develop mammary tissue at different points along this ridge. Well, in humans, in both males and females, a bit more obvious in females after estrogen is released at puberty, breast tissue and nipples can develop anywhere along this mammary ridge. And so humans can be born with extra breast tissue and extra and nipples along this ridge. And there are other examples as well. Even though as adults our red blood cells lack nuclei, as embryos they possess nuclei, just as the condition would be in reptiles. Just as primitive vertebrates are obviously segmented, forming these blocks of tissue known as somites, so too do somites form in our embryos. So there was one more. Just as the eye, the retina, actually forms as part of the brain, we can see that the retina forms as part of the brain, which then stretches from it in the development of the brain. Our hindbrains have a segmentation which is not present in adults. And so there are all of these traits which our embryos have, which are not expected if the human form appeared from nowhere. If the human form appeared from nowhere without connection to other organisms, then embryos would have no expectation other than to go from a simple state directly into the adult human state. These developments of traits which we don't find in adult humans, but which we associate with more primitive mammals or reptiles or amphibians or fish, then which have to then be undone to form the human form. This is A, unexpected, but B, this increases the vulnerability to birth defects. So for example, when our pharyngeal arches form here, the material which ends up being the thymus forms high and the thyroid low. But then these switch places, all right? And as a result, some patients then have birth defects where there's abnormal masses of thyroid and thymic tissue from these different positions. So if one were to propose models which explain, aspects of biology, that's wonderful. And it's wonderful to doubt and to question and come up with new models. But then the question then is, well, then, we don't end with the model. You don't have a hypothesis and now let's teach us in the schools, for example. You then test that. And the creation model, which holds that we are 100% unrelated to other organisms, simply doesn't explain why so many of our embryonic features do not resemble the adult human form, but instead resemble the form of other animals. Intelligent design, claiming that structures cannot, evolve in stages, simply does not explain how, for example, an eye can seem to be a specialized region of a brain which was already photosensitive, or that vertebrae could form from a notochord and cartilage which develops around it, or that a heart could develop in gradual stages. But the same gradual accumulation of changes leading to complex structures, which we get from anatomical and fossil analysis, we can actually see in humans as small bones in the skull fuse to make larger bones, etc. And so the embryological analysis strongly supports the evolutionary model and strongly contradicts other models. Saying otherwise simply does not represent the study of embryos.